Hello, welcome to our webinar today, our SURF webinar. Just a bit of info for those who are not familiar with SURF. SURF stands for the Sustainable Remediation Forum. We're a nonprofit organization dedicated to advancing sustainable remediation practices with members from industry, government, regulatory agencies, consultants, academics, and as well as students, just to name a few. You can go ahead and go to the next slide. For more information, you can visit our SURF website. We have white papers, case studies, links to webinars, links to training and sustainability tools, as well as much more. While there, go ahead and enroll in our newsletter. Make sure you receive our monthly Surf Break newsletter, and check us out on our social media, LinkedIn, Twitter, as well. Today, we're excited to have a great presentation by Intrinsics Environmental, speaking about endophyte-assisted phytoremediation. We'll be focusing on new tools in phytoremediation and associated remediation benefits. Our two speakers are Renee Murphy and Galen O'Toole. Renee is an environmental scientist with Intrinsics. She has a master's in plant science and agriculture from Cal Poly, Pomona, and a bachelor's in business from USC. Renee is assisted in installation of groundwater, soil, and phytoremediation methods with regulatory approval that have resulted in shutdown of numerous mechanical systems. Renee is presented on many platforms, including fairly recently a keynote speaker at the Nicola during the Resiliency, Nature and Climate Solutions Conference in South Africa. Galen's a leading environmental engineer and project manager with Intrinsics. He holds a bachelor's of science in environmental resources from Cal Poly Humboldt. Galen has worked as a project engineer in developing water treatment technologies and potable water reuse, wastewater, desalination, to name a few. In his role in Intrinsics, he designs and deploys endophyte-assisted phytoremediation systems for contaminated soil, groundwater, and surface water at a variety of sites and clients. So with that, I'm going to hand it over to Renee and Galen. And just a reminder to folks, we have AEHS West coming up. So if you like what you see here today, make sure if you're there to check out what SURF has to offer, as well as Intrinsics will be there. And with that, Renee and Galen, I'll let you take over.
Before you start, Galen, I just want to give a shout out that Galen will actually be speaking at AEH West. So go check it out if you get the chance.
Thanks, Renee. And thanks, Chris, for the introduction. Our company is located in Silicon Valley, California, and where we have assembled a diverse team of interdisciplinary experts focused around phytoremediation. For the last, I don't know, I think the team has been working on this for more than 20 years, but this particular approach has been developed for about the last 10 years, where we've been bringing microbiology tools into phytoremediation and creating fully engineered solutions for diverse contaminated sites in all climates across North America. We've been adapting our technology to each site's unique challenges and to create standalone treatment solutions, as well as integrating with a wide variety of other remediation approaches. So just to go over a little bit about what we're going to talk about today is first, if you're not familiar with phyto, we'll have a brief description of phytoremediation and its benefits, and then talk about recent developments in phytoremediation technology and our technology specifically, and then we'll go through a few case studies. So Renee is going to take it away.
Hi, everybody. I'm Renee Murphy, and as an environmental scientist for Intrinsic Environmental, I want to show you how phytoremediation works with our new technology. I also hope to inspire you to look at phytoremediation in all of your feasibility studies and talk with your stakeholders about the benefits that phytoremediation offers. When compared to conventional remediation, phytoremediation is lower cost, has a lower carbon footprint, it can increase land value, it is sustainable, regulator approved, and I think what's not talked about enough is the beneficial social and community aspect. But for
Next
those of
slide.
you, oh
Sorry.
yes, next. And for those of you who aren't familiar with phytoremediation, here's a quick refresher. So phytoremediation is the process of sunlight, transpiration, and photosynthesis. So transpiration is the pump, and photosynthesis provides the fuel for the process. Now, if you look at this image, notice how substantial the root system and the rhizosphere is when you compare it to the tree. This rhizosphere is where degradation of contaminants occur because of the symbiotic relationship with the microorganisms. Microbes, these are found in huge numbers in almost every environment on Earth. They actually colonize the internal and external tissues of plants in a symbiotic way. And very similar in humans, when we have a healthy gut, we're healthy as humans, and trees are the same way. With a healthy biome, trees are healthy internally and externally. So let's talk about a little bit more about how the tree microbe relationship fits into phytoremediation. Next slide, please. So early phytoremediation efforts without intentional application of microbes were successful, but not always so consistent. So if you look at the diagram above, this yellow plume represents the contamination. And where the areas are exceptionally high, you notice that represented by the red dots, those are dead trees. The contamination kills the trees from phytotoxicity. So this, in fact, just reduced the effectiveness of phytoremediation. So let's look at how microbes can benefit phytoremediation. Galen, sorry.
Yeah, thank you. So our company and our founders were involved in some of those early efforts in phytoremediation and noticed these drawbacks, and so started working on what are the extra tools that we can bring in? How can we integrate phytoremediation to be more resilient? And more than 15 years ago, they started working with Dr. Sharon Doty at University of Washington. And she has been, she has been researching the tree microbiome for over 30 years. And her approach has really been to go out into the field, into really sometimes hostile or depleted environments, and find trees and plants that are thriving despite, say, being on a contaminated site or having super low nutrients or being in some other less amenable growing situation. And she's sampled the tree microbiome, hundreds and thousands of distinct strains of organisms per tree, and then really focused in and been able to isolate the 2 or 3 endophytes, the 2 or 3 microbes that are really helping that tree survive those particular challenges. And so, next slide, please. So we are now able to take those endophytes that she's isolated and inoculate them, reintroduce them to trees and plants that we plant, or that we introduce to similar growing conditions, similar hostile growing conditions at other sites. And as we know, contaminated sites have usually multiple environmental stresses, whether it's salt or heat or a variety of contaminants. And so these these endophytes, these bacteria, allow the trees to thrive in the presence of pollutants, in low nutrient areas, in poor soil, in heat and drought conditions. And they help the trees with biomass and water use efficiency in the process. Next slide. And specifically, we have different sets of microbes that are oriented to different classes of contaminants. So we have a set of microbes for oil and gas compounds, for petroleum compounds, one for PCBs, one for explosives, and a really effective microbe for chlorinated solvents. It's different than anaerobic biodegradation that we've seen in bioremediation in the past. It uses an aerobic pathway, and it fully degrades TCE and its daughter products. And we know this through field testing and lab testing, but this microbe actually uses TCE as its preferred carbon source. And we were first able to deploy this here in Silicon Valley on some of the legacy chlorinated solvents, groundwater plumes from our chip manufacturing and the big tech industry in Silicon Valley. And Renee Murphy is going to talk to you about that first project next.
So this was our first site at NASA Ames. Dr. Freeman, our founder, he's the CFO of our company. He worked for NASA Biological Sciences, and he had the unique opportunity to pilot our very first project. So this was a collaboration between NASA Ames, the EPA, and Dr. Doty of the University of Washington. And again, as Galen had described, the MEW Superfund that existed outside of the NASA Air Force, or the NASA Moffett Field Air Force Base was actually migrating to the base. And the idea was to put in a phyto barrier. And this picture really describes what happens when you look at trees that are not inoculated versus trees that have been inoculated with the TCE endophyte. So you can see the two control group tree lines. They are showing signs of phytotoxicity. They're yellow, they're experiencing chlorosis. And if you look at the inoculated tree lines next to the control trees, they have a bigger biomass, they're green, and they're healthy. So you can see very well the TCE tolerance that's being expressed here. Next slide, please. And just a little graphical representation to show you between the first and the second year growth results. Again, the green bar shows the inoculated trees resulted in higher, larger tree diameters when compared to the control group. Next slide, please. And then once again, year to year, the down gradient wells were reading non-detect when compared to the up gradient wells. If you look at that first column, you can see the contaminants from the up gradient wells, and then again, the down gradient wells reading non-detect. Next slide, please. So ultimately, this is what the site looked like 7 years later, and that's Dr. Freeman there standing in front of the trees. These are 7 years old, and they stand about 40 to 50 feet tall. What was so great about this particular project is that it did effectively cut off the TCE plume migrating onto the base. And the EPA was so happy with the results of this project that there's a future housing redevelopment project planned for this site next year, and the EPA wants the same trees to be installed once again to keep that area safe from the plume as well. So that was an example of our TCE endophytes, but Galen is going to show you how we can also deal with oil and gas contamination. Next slide, please.
Thanks, Renee. So this site is in Central Valley. There's an old diesel and gas transfer station with leaky storage tanks. Pretty common scenario across the country, or the world, actually. And it's an example of where conventional remediation was failing. Pump and treat was in place, and soil vapor extraction were in place to address the free product floating on the groundwater plume, and to address vapor intrusion issues into neighboring houses. And it was failing because it was just clogged and high maintenance and high operations costs each year, over $1 million of operation and maintenance for that system. When we got to the site, it was bare, but the smell of fuel was still present when walking the site, and it was very, very hot. Next slide. So phytoremediation had been implemented before we got involved, a small test plot. And as you can see in this photo on the left, a lot of the trees were not doing well, were dying, or showing severe stress. And you can see the black dots are trees that are dead or dying, and orange and red are trees that are stressed, green are healthy trees. So this map is actually almost a perfect overlay of the plume map, and gives you an idea of the relative concentrations on site. The groundwater here is shallow, so the trees were able to reach groundwater relatively quickly, about 10 feet down. And so the first thing we did was inoculate these rows outlined in blue with our oil and gas degrading endophytes. And you can see on the right-hand picture the effects. The row on the left is inoculated trees, which are much taller and greener and larger, and there's fewer gaps in the line than the row on the right, which are uninoculated control. So this success gave the client the confidence to implement phytoremediation across the entire site. And so we came back and we planted a combination of salt-tolerant poplars with our endophytes across the whole site, almost 1,000 trees. And this, was quickly successful, and the trees went from high mortality rate to over 90% success establishment rate. Next slide. And the effects for treatment on site were readily apparent. So this is a graph of natural source zone depletion rates over time. And the yellow line on the bottom shows the natural source zone depletion rates out of the phytoplantation, off the as represented by CO2 vapor analysis, and gallons of octane per acre per year. And so there's some seasonal fluctuation you see, but it's pretty low. And in the phyto plot, in the root zone of the trees, the degradation is much higher and increasing over time as the trees mature. Fortunately, we don't have more recent data, but this trend continues upwards over the last 2 years. Next slide. I mentioned that this was based on CO2 data. This work was done by ETIC and eFlux. And this shows a map of soil vapor, soil CO2 concentrations across the plot. So in the root zones of trees, we see much more CO2 production, which, as Renee was showing in that early slide, the trees and the root zone really reestablishes a healthy microbial community that helps directly to break down, as well as the trees, both breaking down and controlling groundwater. But the proportion in green is petroleum-derived carbon, and the proportion in yellow is atmospheric-derived CO2. So both the total amount of CO2 produced, indicating soil microbe health, and the proportion that comes from degraded petroleum is much higher in the root zone than outside of the planted areas. Some of these circles are so small you probably can't even see them when they're outside of the root zones, outside of the planted area. Next slide. And so, one of the primary mechanisms that we use on sites with the trees is to control groundwater. And healthy trees that are growing fast and large can transpire a lot more groundwater, thereby creating a cone of depression in the water table and preventing further migration off-site. And we see that here in this graph as a separation between the green and the blue lines. You see on the very left hand, when they're before the trees, or soon after the trees are planted, there's still some lines are not fully separated, even though the pump and treat system is operational. And then over time, as the trees establish, the separation between the in-plot and out-of-plot wells elevations grows. And so within just a couple of years of planting, the State Water Board authorized the shutoff of the groundwater pump and treat system and the soil vapor extraction systems, as the tree plantation was showing better, more effective remediation and groundwater control in this area, which is effectively source zone treatment for this site. Next slide. And now, what used to be a community eyesore and source of vapor intrusion is becoming a community forest. And the local residents have mentioned that they really enjoy having this site across the street now. Even there have been requests to take down the fence so that they can enjoy the trees. But it is ongoing remediation, with now site closure contemplated in less than 10 years. Next slide. Oh, and I can't forget on the previous one, the client is saving a lot of money, approximately $1 million per year in deferred maintenance, as the trees do require maintenance, but not on that scale. Next slide. So this site in Corpus Christi, Texas, was similar inside a refinery, but a transfer station at a refinery. And spills over time. The site was brownfield, unable to grow or support even weeds very well, and with also the smell of fuel present. The primary contaminant here is benzene, and we co-planted with our hybrid poplars, as well as a deep-rooted native grass that we inoculated with the same endophytes, the petroleum degrading endophytes. And you can see on the far right, after just a couple of years, about 2.5 years, we see healthy trees, a lawn, and next slide. And a steep reduction in benzene concentrations in the middle of the plot and down gradient wells. So outside of the plume are the monitoring well, the green line at the bottom and the orange line at the bottom are outside of the plume. The red and blue lines are showing wells in the middle and down gradient edge of the site. And both show significant reductions installation was here in 2018, and by 2020, the concentrations are getting much closer to the regulatory threshold, which is down in the dotted line at the bottom of this graph. And Renee was just in, she just came back from Alaska, where she presented at the Alaska Forum on the Environment. And we have a site up in Fairbanks, Alaska, that she's going to discuss next.
Great. Next slide, please. So one of the questions I get asked fairly often is what type of climates can we work in? And Galen just showed on the last slide how we worked in a hot and humid environment. And as you can see here, we can also work in an Arctic climate. So this was in Fairbanks, Alaska. It was a transfer station. So fuel was brought in from the railroad. There were holding tanks and a truck loading area. What complicated this site was that it was a mixed waste site. So it had a chlorinated solvent plume as well as an oil and gas plume. And in this picture, you can see the hot spot of the contamination is in the center here. There is an active air sparge system, which is combined with our phytoremediation system. And what we did on this site was that we augured about 7 feet deep through the asphalt, and we used 12-foot tall cuttings. In the foreground, which you can't see, we trenched trees in to help remove benzene from underneath the road. Next slide, please. So we are having very positive results on the site here. The results aren't yet 100%. There are going to be, there are proposed a few more trees planted for the upcoming season. A couple of things I did want to point out are these images are showing pretty consistently what the site looks like across the board. The trees are doing really well, and on the right side you can see the air sparge system, and you can see that the trees are doing really well, even though that is technically one of the hot spots. So this remediation is an ongoing effort right now. The trees are degrading the contaminants and they are surviving well. And the regulators are pleased with the process and continue to watch, and hopefully this will allow us to do a lot more work in Alaska. Next slide, please. So this site here, it's a TCE contaminated site in Danville, Illinois. It was actually designed after the success of the Fairbanks site. This was a former manufacturing facility that had high concentrations of TCE, DCE, and vinyl chloride, about 7 to 15 ppm, just to kind of give you a ballpark of what was going on here. And Intrinsic designed a system of strategic plantings to exert hydrological control over the plume. We planted 600 hybrid poplar trees inoculated with our chlorinated solvent degrading endophyte. There are a few things I want to point out in these pictures. If you notice that skid steer at the top picture, that shows how low impact our installations actually are. It took about 2 days to auger the holes in and to plant the trees. That's it. That is all the equipment that we're using. Now, if we're looking at sites that are more remote and we don't have access to a skid steer, trees can be hand augered in. So if you could imagine what conventional remediation efforts look like versus phytoremediation, there's just such a low carbon impact. And if you can imagine, there's a community around this area as well. So I just like to point out ultimately what this site looks like to the community. You can see the picture on the far right where we have this lovely forest of trees and we've got the rainbow going overhead. I promise I didn't Photoshop that in. But you can see the site is beautiful. They are looking towards future site closure, and the city's also turning this area into a park. Next slide, please. I also want to show you how phytoremediation can be used as a redevelopment tool. This is a brownfield site that is in Playa Vista, California. It's an old Howard Hughes aerospace facility that was redeveloped into beautiful corporate offices. But what I like to point out about this site is that we were able to remediate during the development process. So it was actually incorporated into the overall landscape design as a functional landscape. So ultimately, the risk, the liability was eliminated over time. And go ahead, next slide. I want to show you the results of this project. This is an outside view of the functional landscaping. It took about 3 years for the 6-foot cuttings to become these adult trees that were providing shade, decreasing the heat island effect, and ultimately added beauty to this campus. Specifically, we use salt and boron tolerant hybrid trees for this location because of the brackish recycled irrigation water and the proximity to the coast. I do want to talk a little bit more about the fact that, phytoremediation, it is a biological system. And our engineered systems do have a unique project cycle. Next slide, please. So, and as the trees have their year cycle, we need to coordinate our project cycle. So this project site assessment and conceptual design is optimal for spring to early summer with regulatory buy-in as soon as possible. And because a detailed design and authorization needs to be completed by the beginning of winter, we also would like to have our tree reservation needs to be done between December and January in order to install the site in March through July. And again, there's a little variation there, just depending on the site, on the climate of the site. Next slide, please. We've been really lucky to work with all of these incredible partners and collaborators, and without their help, we owe our success to the help of these different partners. Next slide, please. We'd love to answer any of your questions, and we want to invite you to imagine how endophyte-assisted phytotechnology could be applied to any of your future projects.
Renee, yeah, thank you. Just a reminder to all, we've been seeing some questions coming in already on the questions chat, but if you have questions, that's how we're working through those. So go ahead and drop those in the chat, but we'll go ahead and get going. So I guess first question to you guys, during your study, do you identify differentiation in phytoremediation capacity between the inoculated trees and the controls?
Yes, it's a short answer. Are you specifically referring to the NASA Ames example that on the MEW plan that we discussed, or just in general?
I think somewhat in general, that's about the timeline that question came in to us. So
okay. So we use a variety of what we call phytoforensics tools to show efficacy and show difference in treatment. So typically on a site, we inoculate all the trees because we are providing an effective solution. But on some of our early sites, we have uninoculated controls. And so how we tell the difference between effectiveness is through a few different mechanisms. We first and foremost, we will measure tree tissue VOCs, and through tree core analysis or lower branch analysis using a GCMS headspace method. And this tells us, how much the tree is uptaking. And what we see is that in inoculated trees, the concentrations are, 1,000 times, 100 times, 10,000 times lower than uninoculated trees, because endophytes are degrading the contaminants in the tree trunk. And then we can measure also specifically for chlorinated solvents, the salt content. And so as TCE is broken down, chloride is released into the tree, and the trees shed that by fluxing it out of the roots into the soil and concentrating in their leaves. And so we measure the leaf ions to show, and we measure much higher chloride and leaf ions for inoculated trees on chlorinated solvent sites. And then when we measure soil chloride, it's also elevated over controls.
Yeah, so I think similar, I kind of answered this, but when you collect those tree cores, like from the less sites, do you collect those to verify the hydrocarbons are in the trees, like in the control trees, or?
It's typically for a couple different purposes. One, it shows that there's uptake. So, we can't quantify the uptake because the endophytes are degrading the hydrocarbons and VOCs so quickly. But what we can do is we can prove that there's uptake. So that proves that the roots are in the groundwater in the zone that we want them to be, and that they are drawing up the contaminated groundwater versus getting their water from irrigation or the soil or some other source. And so that step is a verification, and then when we monitor the ions, then that's really more of an indication of breakdown for chlorinated solvents for oil and gas. We rely more on the soil CO2 analysis to show that there's significantly more degradation happening in the soil as we reestablish trees and the soil microbiome.
The next two here, kind of, I think, similar lines of questions here. Are all the isolated endophytes used for your pilots taken from the University of Washington study? And then kind of as a tie into that, do you culture the endophytes at intrinsics? If so, do you have a library of different endophytes for different contaminants and site conditions?
Those are really good questions and spot on. We have most of our endophytes from the University of Washington, but not all. We're working on acquiring others that could work in tandem. And we do a lot of R&D to make sure that the endophytes we inoculate with work well together. So, there's not adverse competition between the ones we're inoculating with. And, especially for mixed waste sites where we inoculate with multiples in the same organism. And yes, we do keep a library of endophytes that are tuned to specific classes of contaminants, and we culture those and keep our strains isolated at intrinsics, and work on doing the small trials we need to for R&D, as well as growing those out for installations.
So I see.
Thanks.
Sorry, Chris, I see a couple of the questions coming in. I'm sorry, I didn't specify that at the Alaska site, we actually use balsam poplar. Someone had asked if we had used cottonwood. And Galen, have we worked with coniferous trees at all? Someone was suggesting it'd be a more effective year-round in northern latitudes.
We have worked with some and done research with some of our partners with coniferous. I don't believe we have installed a coniferous-based site yet, but there are several conifers that are in our palette of plants and trees that we have ready to use on sites where they're appropriate. And we have done testing to show that many of our endophytes work in conifers as well, and that they can continue to maintain and increase their population in those trees as well. So, that brings up another good point, is that we, one of the things that our founder, Dr. John Freeman, and our partners at PPCU have bring to our interdisciplinary team and our technology is a really in-depth understanding of the plants and trees that can be used for phytoremediation in a lot of different environments. And so we have a very wide palette of both trees that we can introduce and native plants and trees that we can use for most climates in North America and abroad.
Yeah, and I noticed there was a question someone asked, I'm trying to find it right now, regarding native species. So some of our sites, we do incorporate native species, and it really just depends on what we're trying to achieve. Sometimes we need to bring in salt and boron tolerant hybrid poplars. Sometimes we're in an urban setting and there's requirements for the types of trees that we're using. We really like to use the poplars because their applications are very useful in city type atmospheres. We can plant them very close together. We can prune them up. We can plant in containers. They work really well in a pump and treat type system. Oh, there's the question on the natives. So yeah, it is very site specific what species that we are going to use, but native trees are always something that we're looking at, and if not blending it into our overall design.
And I'll actually maybe.
Yeah, go ahead, Chris.
I was going to just chime in there too. I know I was involved with the Fairbanks site, and on that site we used a balsam poplar, which was a native tree to Alaska, and part of the reason that we utilized that tree instead of the hybrid poplar was sourcing, but also availability, its resiliency in the cold climate.
Yes, absolutely.
So, while hybrid poplars do have relatively short lifespans, we use them for a few reasons. They just transpire so much more water than natives. So where we're looking to control groundwater, they're really valuable, either primary or component in a mix of species. And then the fact that they have short lifespan can actually be beneficial on sites where restoration to native species is a goal, because they will, they're sterile, they're cottonless, they won't propagate, and at the end of their life cycle, they quickly give their nutrients and the carbon they accumulated back to the soil and the native species that are taking over. USDA Forest Service has been working on protocols to actually use hybrid poplars as a nursery species in restoration efforts. So planting natives and poplars concurrently in restoration, so that the poplars will shade out invasives, but the natives are better coped with growing in their shadow and then taking over after a few decades as the poplars wane in the system.
Yes. There are also a lot of questions, Galen, coming in about maintenance of the tree, how waste management on pruning these trees is handled. One thing I'd like to clarify is when we're working with organic contaminants, the degradation is actually mostly happening within the rhizosphere and within the trunk of the tree. So it's not technically, I guess, the visualizations that it's being stored up in the leaves. So when we're pruning these trees, that they are, contaminated or toxic in some way. That's more the case when we're dealing with inorganic contaminants, such as heavy metals. The trees are uptaking the heavy metals into their system and storing them in their biomass. And if we were working with contamination where we were looking to deal with heavy metals or inorganic contaminants, we would design a system that would be different than, say, planting these poplar trees, because they're degrading the contaminant differently. And we would have to look at part of the maintenance being pruning or taking the biomass of the plant and disposing of it properly, hoping to trim and cut the plants back when they're still under the hazardous waste levels for disposing. But that would be integrated into the system if that's what we're dealing with. But when we're looking at the way we're dealing with oil and gas and TCE, we're not having to worry so much about pruned trees or pruned arms and leaves and that type of thing, because they're not storing that contamination. I hope I answered that. And Galen, you want to?
Yeah, let me chime in a little bit there. We have done some testing on composted mulch from from some of our sites' pruned material, TCE, 1,4-dioxane, and others. And each time it's come up non-detect. So once those limbs and leaves and whatnot are harvested and chipped, the concentrations are very low. And as Renee said, metals accumulation can be an issue on those sites, but we would design a system very differently for metals extraction than for VOCs, whether petroleum or chlorinated solvents. And there's a bunch of good questions here. I think we touched on the waste management a fair amount, but
Galen,
yeah.
do you want to talk about, I think there were some questions about how far the tree roots can go, and talk about pumping.
Yeah, I see those two based on groundwater and beta zone. I want to get to those in a second. I just want to make one more point on maintenance, which is, we mentioned earlier that the phytoremediation is low maintenance, but not no maintenance. So the trees are really the key to the system in a couple of ways. And so keeping the trees healthy both requires endophytes, which are bacteria and other microorganisms that live inside the trees. We can inoculate them, but then they move, by spraying it on or dipping the cuttings or pouring some inoculum in the root zone of a rooted plant or tree. But then those endophytes, once they make it in, move all throughout that organism, both into the branches and the leaves and down into the roots, and just take up residence there. And we can, when we've measured, sampled for their presence years later, they're still present and doing their job. And so part of the maintenance is, maintaining healthy population of endophytes, maintaining healthy trees, and that's can be some pruning, irrigation on some sites, treatment for pests, that sort of thing. That's the primary type of maintenance, and it doesn't amount to typically a very large budget year to year. And so, they're treating the tree roots, especially let's use hybrid poplar plantation as an example, or any tree plantation. They'll be extracting groundwater for VOCs. The endophytes will be breaking down those inside the tree, and then so that groundwater is treated, that is taken up, but also in and around the root zone, both in the beta zone and in the saturated zone, the roots can enter the groundwater, up to 5 to 10 feet, depending on the site. There will be additional microbes living outside of the roots, in the rhizosphere, that will be benefiting greatly from the tree's presence, because they push a lot of nutrients into the ground and foster these microbial communities outside of their roots. So the breakdown doesn't happen just in the tree itself, in the groundwater, but also in the soil and the upper surface of the water table and the capillary fringe, in the native microbes whose populations boost dramatically once healthy trees are again on the site. And so the tree roots can reach down up to over 30 feet to reach the water table, as long as there aren't any, layers of bedrock or significant impediments to their descent. And where there are, we can bore down with boreholes right to the water table and give the roots a clear passage to come straight down and reach the water table more quickly.
Galen, there's another question here. Can you describe a site that we worked with PCBs?
Sure. We currently have done pot studies with PCBs, but we do not have a fully installed site with PCBs. And so we would be looking for a first site. We have microbes that are shown to degrade PCBs. They live both in the root zone of both inside and on the surfaces of the roots. So we know that PCBs are very hydrophobic, so they're not going to be drawn up into the trees or plants readily. And so the degradation takes place more at the root surfaces as PCBs are drawn, mobilized and drawn to the root surfaces, but then excluded from the water and other nutrients the plants and trees are uptaking. And this strain that we have is very closely related to, it's been fully genome sequenced, and it's 99.8%, 99.9% similar to LB400, if you're familiar with the one that Gerald Schnorr identified on switchgrass. And our CSO, Dr. John Freeman, is reminding me, because my memory is fading, I guess, that we do actually have a PCB site. I actually installed it 2 years ago, so I should know this.
That's okay.
The primary, it's a Superfund landfill in upstate New York. And there's two primary contaminants, PCBs in the landfill and TCE in the groundwater underlying landfill. And so the trees we installed are for controlling the water table inside the landfill and drawing up water, keeping the groundwater table below the outside water table, so it's a contained area. And the PCB degrading strain is inoculated in those trees as well to help protect the trees from those the toxic effects in the soil as the roots grow down. And that site actually is one of our hottest sites. That we had, last I checked, 100% establishment, but the TCE concentrations are up to 35 ppm. So it's a really, really hot TCE site, but the trees are still establishing and growing well.
And Galen, I don't know if you read this incoming question. That's pretty technical. You might want to read it.
That one would be for, I'm going to read this one out loud. Maybe Dr. John Freeman can send me some ability to answer this one, because this one's outside of my knowledge of microbiology. I am an engineer that has studied nature-based solutions for treatment, but not this. So this one is, have you tried to identify the biochemical pathways being used by the endophytes for chlorinated aliphatic degradation, e.g. via certain monooxygenases or cytochrome P450s? So we know that hybrid poplars have a P450 enzyme that they use to degrade chlorinated solvents. We don't know precisely the metabolic pathway that is used for degrading chlorinated solvents by the endophytes, but in bench scale, Chris, in just a second when I finish, maybe you could unmute John and he could answer this one. But we have done, Dr.
I'll see if I can.
Did studies with TCE and the PDN3 endophyte showing that there's a molar ratio of chloride produced in 3 to 1 as TCE is degraded. So we know that the endophyte is fully stripping chloride off of the ethene backbone and using it as carbon source. And there are no, at the end of the day, there are no degradation products left over. It's this new aerobic pathway that hasn't been fully explored.
And you nailed it, Galen. For a non-microbiologist, I'll say, very, very well done. It's an aerobic pathway. We don't know much about it. We don't see those normal monooxygenase dehalogenation type genes. The P450s are in also the trees, and so that is helpful. But the microbes themselves do use it as a carbon source, and so they strip all of the chlorides off and use the ethene as food. Yep.
Thanks.
Yeah, thanks, John. Yeah, I think with that, I think we've hit on all the questions that we've had come in. I really appreciate all the questions that we did receive. So I guess with that, we'll just go to the next slide here, Amy. I just want to touch on a few things with SURF. Just remind everybody a few ways to get involved with SURF. We have the Remediation Journal. We have these webinars for SURF members. Go ahead and reach out to us if you want to get involved. Presenting webinars, helping us find interesting topics to share. We also have our technical initiatives. We have a current TI related to environmental justice going on, so reach out if you'd like to be involved in that. Next slide. As always, we do want to thank our SURF sponsors. Without them, none of this is possible. You can reach these SURF YouTube. You can reach us on our SURF YouTube channel for this webinar as well as past webinars. Watch out for upcoming future webinars. And then again, we'll be at the EHS West conference with the SURF technical sessions, our SURF open meeting. And I guess with that, Galen and Renee, thank you for presenting today. And thank you to TriHydro for supporting us behind the scenes and running the webinars for us. Thank you, and we'll look forward to seeing you on the next one.
Thanks. Will there be a recording available later?
Yep.
Awesome. Thank you.
Thank you.